Okay, go ahead.
All right, it's my pleasure to introduce Camilo. They're a Colombian biologist specializing in aquaculture nutrition, concentrating on the physiology and nutritional aspects of freshwater fish. His primary interest lies in uncovering the intricate relationship between gastrointestinal ecology and animal health, while also exploring the practical application of functional feed additives and nutraceuticals. Take it away, Camilo.
Okay, good afternoon, everyone. Thank you so much for your introduction. As you told you, my presentation is in vitro assessment of carbachol, thymol, and citral, exploring their potential to inhibit fish pathogens. Well, like as all my colleagues speaking of this day, catfish is one of the most important fishes for the aquaculture production here in the U.S. Nevertheless, have the problem that is really vulnerable to animals like Aromonas, Edwardiella, or Streptococcus. We can use a lot of different antibiotics. Nevertheless, the State Food and Drug Administration let it use just terramycin, romanet, and apoflor. And even with that, we can use a lot of these ones because have antibiotic resistance. With that in mind, the catfish nutrition aquaculture here in the Mississippi state proposed the use of phytocompounds. Phytocompounds are elements that are produced in the second metabolisms in the plants, and they have a lot of properties like antimicrobial, immunostimulant, antioxidant, growth promoting, and anti-stress properties. One of these compounds are really proven in different other productions like cattle, poultry, or pigs. You have two for agriculture, like the tilapia and trout. Nevertheless, one of the problems of these different studies is that these studies used the essential oil in there totally. And that is a problem because these essential oils is a mix of many substances that could be affected the composition for elements like the weather, the weather, the methods of extractions, the different kind of species of the plants. So with that in mind, we decided in the aquaculture nutritional lab uses the main components of these plants. One of the plants that there is more important for this kind of research is the lemongrass, the thyme, and the oregano. And the main components of these plants we can find at the citral, the thymol, and the carbachol. With that in mind, this study object was evaluating inhibitory effects of the phytocompounds citral, carbachol, and thymol on fish pathogenic bacteria. We consider that doses lower than minimal inhibitory concentrations can inhibit biofilm formation and reduce hemolytic activity. For that, the methodology that we decided was select a different pathogenic bacteria that was isolated in outbreaks here in the US. You can find different elements like Streptococcus, Aromonas, or Edwardiellas. And we expose all of these different bacteria to concentration of the citral, thymol, and carbachol. We decided to use 4 different experiments in this research. The first one of them was the Kirby-Bauer diffusion susceptibility. You can see here more or less all of the steps that we follow. In this test, the idea is determine if the bacteria are susceptible or not to the presence of these components. At the end of the results, we're going to have the diameter of the halo. The second test was the Brock microevolution method. In this method, when we know if the bacteria are vulnerable or not to the presence of components. Sorry. Okay. We are focusing if we need to know the concentration that these bacteria are vulnerable. So in that way, we're gonna follow all of these steps to determine the minimal inhibitory concentration and the minimal bactericidal concentration. The third test was the antibiofilm activity compounds. As you know, the biofilm formation is one of the main steps to start to viral factors, for the bacteria start to express viral factors. So that's why we select. We want to know if these bacteria could be affect this biofilm formation for the presence of the phytocompounds. That's why we repeated this. We use this methodology. We expose the bacteria, the different cultures to different concentration of the phytocompound. The final assay was a hemolytic assay. Many bacteria, in this case, the bacteria that we use it, the strains have a production of hemolysis. Hemolysis are exotoxins that produce the bacteria to attack the red blood cells. That's why in that case, we follow these steps that you can see here in the screen and expose the bacteria to different concentrations of the compounds, incubated that. And after if that incubation, that was the time that the production of the hemolysis, we take off all of the bacteria and expose that for red blood cells to check the hemolysis present in the mixed. This is our results. You can see here our diameter of the halo, the different bacteria, and the different compounds that we evaluated. You can see that in all of the cases have a significant difference in this part. In the part that I select right now, you can see, for example, that the elements like a thymol or the carbachol have a really similar behavior, and the bacteria are more vulnerable to the presence of these components. If you compare, for example, with the citral, we can understand that because if you analyze the structures of the molecular structure of these compounds, you can see how, for example, the citral is a linear molecule that propends to be more vulnerable to different elements like a degradation. In the other hand, you can see that the citral have a high electronegativity, have a polarizate that they're going to influence in the interaction that have this kind of a molecule. And you can see, for example, too, that, for example, the molecules of the carbachol and the thymol are really similar between them. That's why they have a similar behavior in the Kilby-Bauer test, diffusion test. And on the other hand, you can see, for example, the only difference is the presence of this, the position of this hydroxyl group. These two molecules have the protection additional that the presence of the benzoic ring, that ring provides more protection or more stability to these kind of molecules. In these pictures, you can see the different halo diameters that you can find. So the control with respect to the carbachol, the citral, and the thymol, and we can see that this is so similar with the inhibition halo for other antibiotics, like, for example, like a chloramphenicol or tetracycline in this one. This is our results for Brock microevolution method. In this table, the important is that you can see the different strains that we evaluated, the minimal inhibitory concentration and minimal bactericidal concentration for the three phytocompounds. All of these concentrations are evaluated and back up for different other studies, for example, like weight, acil, Kumar. Nevertheless, you can find other studies that contradicts these concentrations, and the reason that we can find is because these other studies had different delivery systems, for example, like nanoemulsions, microemulsions, or even they use a different essential oil in their totality. In this picture, I want to do a little summary of what is the possible reason that these compounds could produce mortality in the bacteria. You can see, for example, the studies of Lambert 221 that decide that interaction with the biliary membrane with the elements like a carbachol or thymol can produce a loss of integrity, permeability in the leakage of material, and lipid bilayer. So we can see here the loss of the different anions. Other studies, for example, like Shi 2016, they demonstrate that the interaction between the citral, this molecule, with the membrane can reduce the pH and reduce the intracellular ATP. And the most important thing, that hyperpolarize the membranes. This hyperpolarization was demonstrated again for the studies of Wei 2021, that they demonstrate that the citral have a high electronegativity, high electric conductivity, and they produce that when they associate with different proteins in the membrane, these different channels of the interface ions, they affect that exchange and that. Finally, you can find the studies of Gao to 2020 that they discovered that the interaction with the citral with the ribosome can inhibit the production of elements important for membrane biosystem, quorum sensing, viral factor, and biofilm. I'm talking about biofilm, about our results. You can see here in these pictures, in these images, that we have found we found significant difference with respect to the control in the most of the treatments. It's important to notice in the arrows that you can see the minimal inhibitory concentration of these compounds. And interesting in this part is that you can find that even concentrations that are low of the minimal inhibitory concentrations have effects in the reduction in the biofilm formation. The example more interesting is that the lowest concentration that we test, that was 24 micrograms per milliliter, can produce an innovation in the body of information. And that is really important because if you can see, for example, these cytotoxics in different cellular models, you can find that cytotoxicity of these compounds is in the order of 2500 or 62 or 18. That means that at the end of this study, we can use these components in the really low in concentrations, can affect the bio of information, but this don't gonna be produce any kind of cytotoxic in the host, in that case, catfish. Other the reasons that we can find that maybe why happen these couple of elements and reduction of the bio of information is because, for example, in this study that when this was exposed, Candida albicans was exposed to the citral, was a reduction in the strength of the EFAS. Or for example, this other study of 2017, that when they was exposed Streptococcus aureus to carbachol and thymol, was a reduction in the population. Respect to hemolytic assay, you can say for the strains that present hemolysis, in that case, Streptococcus ineae and in Aromonas hydrophila, we can find that when we expose these bacteria to low concentration, to, for example, like 12 micrograms by milliliter of the different compounds, we have a significant difference in the reductions in the hemolysis activity. With that in mind, our conclusion is that the bacterial pathogens proposed in this study show sensitivity to the phytocompounds study, that the concentration of the phytocompounds affect bacterial growth, concentrations lower than the minimal inhibitory concentration have effects on bio information, and the phytocompounds inhibit hemolytic activity in the treatment conditions evaluated. As the future directions, we're gonna do an scanning electron microscopy. The idea of this one is describe how changes the morphology with the bio information in these bacteria, made a qPCR to determine what genes are upregulated or downregulated in this bacteria. For example, we're gonna focus in genes like, for example, for bio information or viral factors. And finally, expose these animals to feeding trial to determine how these components can affect elements like elements of production or in the health. I want to thank to all of the catfish nutrition equipment and the data equipment for Barmer Aquaculture Center, Mississippi State, and obviously you for your attention. Thank you so much. Any questions?
Great job, Camilo. Thanks for the presentation.
Thank you so much to you.
Yeah, folks, if you want to unmute yourself, go ahead. We have one question here in the chat. They're curious why you used sheep red blood cells instead of catfish or other fish species in the hemolysis assay.
Well, actually, it was a mistake in the presentation because the first time that I present this example was I used sheep red blood cells. Nevertheless, in the second experiment, I used catfish red blood cells. I'm really sorry for this understanding.
Great. Any other questions out there? If not, thanks again, Camilo.
Okay, thank you so much for your